and welcome to the very first episode of Pipetting Hot Tea. I'm Lilia, and I'm thrilled to kick things off with an inspiring guest, Dr. Roberto Chica. He's a professor here at the University of Ottawa in the Department of Chemistry and Biomolecular Sciences, where he leads a research lab specializing in computational protein design and enzyme engineering. His work explores how we can design proteins with specific functions, like creating novel enzymes, biosensors, and therapeutic tools. Dr. Chica earned his PhD in chemistry from the University of Montreal before completing a prestigious postdoctoral fellowship at Caltech. Let's now dive into the science behind the molecules and the minds that are shaping the future of biotechnology. I hope you guys are ready, so let's spill the tea.
Welcome to the first episode of Pipetting Hot Tea. In today's podcast, we're diving into the fascinating world of proteins. Not the kind in your smoothie, but the kind scientists are designing from scratch to solve real-world problems. Our guest today is Dr. Chica, who's at the cutting edge of protein engineering and computational biology. Dr. Chica, welcome to the show. Can you tell our listeners a little bit about who you are and what drew you to this line of research?
Yes, first I'd like to start by thanking you for the invitation. It's a pleasure to be here. So I'm a professor of chemistry here at the University of Ottawa, and my research expertise is protein engineering and design, more specifically computational design of proteins and enzymes. And my background is biochemistry, so my undergraduate degree was in biochemistry, and that's where I learned about proteins. So what really drew me into this field is when I discovered the central dogma of molecular biology that basically DNA contains the information to make proteins, and then proteins actually carry out most of the tasks that a cell needs in order to survive and thrive. That made me really interested and really fascinated that you had these molecules that would do such a wide range of different tasks or functions in a cell. And then as an undergrad, I learned about protein structure and how structure is linked to function. Now, the other aspect of my training that really set me on this path was organic chemistry, which I still teach to this day. But in organic chemistry, what I liked was understanding how you can change the structure of a molecule to give it new properties. So then, I realized you can do this with proteins as well. You can change their structure to give them a new function. And I became really interested in this topic, and I've been investigating it ever since I was an undergrad, did a few internships in research labs, and got exposed to protein engineering in that way.
Perfect. Thank you so much. We also wanted to have an idea, what are some surprising things you've discovered about proteins that they do beyond what you've also learned in undergrad throughout your research?
Yeah, proteins are always surprising me. There's always some new weird protein that somebody discovers that does something you couldn't even imagine before. We learned about the 20 amino acids that make up proteins, right? Mm-hmm. But there's other structures that can appear in proteins where these amino acids react together and they form something new that's not one of the 20 amino acids. It has a different chemical structure, but it's still part of the protein itself. And it gives it a new property. For example, you may have heard of a GFP, the green fluorescent protein.
Yes, from developed from jellyfish proteins, right?
Yes, exactly. So this protein becomes fluorescent because there are 3 amino acids in its sequence that react chemically together to form a new structure that becomes fluorescent. Oh. So when you think about these things, we tend to think of proteins as made up of 20 amino acids, but there's all these other chemical groups that can appear because the protein reacts within itself or with other molecules.
That's true. I wouldn't have thought about that. That's really cool.
Yeah, so the different combinations kind of can create different types of almost like new amino acids kind of? Or
yes.
Okay.
So we wouldn't call them amino acids, but yes, in a nutshell, you're modifying the regular amino acids to create a new structure.
Oh, okay. That's really fascinating. All right, so let's break it down even further and just get super, super basic. What is a protein and why does it matter? What's the whole function of them? Why are they important?
Yes. Okay, so proteins are the types of molecules that perform most of the tasks in a cell. To keep us alive, there are proteins currently doing things in our bodies that are helping us be alive. But ultimately, a protein, what it is we call this a biomacromolecule. So it's a very large molecule made up of thousands of atoms, and they're comprised of amino acids. So if you think of a protein as a chain, each link in a chain would be a different amino acid. And there are 20 amino acids that normally make up proteins. And when you connect them one after the other in a chain, depending on the order in which you connect them together, you get a different protein that will have a different structure and therefore a different function. So what's particularly fascinating about proteins is that given that you have 20 possible building blocks, 20 amino acids, and you can connect them in different combinations, the more you add, the more there are possible combinations. Yeah. So for a protein that has 100 amino acid, that would be 20 to the 100 possible combinations of these amino acids.
Oh, so wow, that's a large number.
That's a huge number. We call it an astronomical number because it's more than the number of atoms that there are in the universe.
Wow.
So what does this tell you? It tells you that there's basically an infinite number of possible proteins that could exist. And that's why, in my opinion, they're used by biology to do things, because there's so many possibilities. You can find proteins for a wide range of different functions, right? For example, we have proteins in our eyes that detect light and enable us to see. We have some in our stomach that are degrading the food we just ate. Yeah. And then you have others in our blood that transport the oxygen from the lungs to the other cells. So there's a wide range of different functions that all come from this class of molecules that are made up of the same building blocks.
Exactly. And as a chemist, from your background, like what made you find protein such a fascinating material to be working with?
Yes, so I became very interested in proteins, of course, when I found out what they did in biology. But given that I'm a chemist, I've always been interested in how things work. It's not so much about, for example, a biologist may be interested in what a protein does, but a chemist will ask, how does it do it? Like, what's the mechanism? Why is this protein doing this, whereas this other protein is doing this other thing? How does it work? And then I've always been interested in creating new things, like building things. So a protein is really nice because there's so many possibilities of modifying them to create a new function, or even now creating new proteins completely from scratch. So we now have methods where we can start doing this.
All right, well then let's kind of dig a little bit deeper into that. So you're talking about engineering proteins. So nature already does such a good job of making these proteins. Why do we need to engineer new ones? What's the importance behind that?
Yes, so there are two aspects that are very important. The first one is we can only fully understand something if we can create it. Basically, if we can create a protein from scratch and it works the way we thought it would, that means we understand it. So there's that aspect that people like to design proteins to demonstrate that they understand how it works. But beyond that, there's a lot of potential applications. So as you mentioned, nature has made a lot of proteins and they're pretty good at what they do. But nature doesn't have the same needs as we chemists wouldn't necessarily have. For example, what if I want a protein that can, an enzyme that would be able to synthesize some molecule that has never been encountered in nature? So nature wouldn't have a need for that because that molecule that the enzyme makes isn't present, right? Furthermore, in nature, the driving force is natural selection, where organisms evolve to adapt to their environment. So they need to be, the proteins need to be as good to enable them to do that, but not better. But maybe we want them to be better. I'll give you an example. Maybe all natural proteins would be stable at normal temperatures, let's say, within the ranges of what is experienced on Earth.
Yeah. Okay.
But what if we wanted to use a protein in an oven, something where we can, crank up the temperature? Nature doesn't need that, maybe, but we do. So can we create proteins that would be more stable? Or one even more concrete example, in organic synthesis, we would want to use enzymes, but often they're not stable in organic solvents because most proteins in nature are found in aqueous environments. So we may want to engineer an enzyme that would be stable under those conditions.
And is this where you apply computational design?
Yes. So computation is really useful because going back to what I was saying about the number of possible sequences,
Yes, that large number.
yeah, it's such a huge number that without computation, it becomes very hard to even, I guess, investigate a small fraction of that. That's not to say that a computer would be able to, evaluate all those sequences that I mentioned. That wouldn't even be possible because there's just too many
Yeah.
possible sequences. But a computer can definitely evaluate more possible protein sequences than a person would be able to do, and much
Yeah, it
faster.
would be very challenging for a person to go through all
Yeah.
of those different companies.
It would be basically a needle, looking for a needle in a haystack.
That's exactly right. So
Yeah.
basically, my job as a protein designer or protein engineer is to find a needle. And to help me find a needle, I develop computational methods to help me search the haystack more efficiently. So instead of going through every single bit
Yeah.
of straw in the haystack,
Yeah.
right, to find the needle, I develop methods to tell me the needle is most likely to be in this area of the haystack. So then what I need to look in is a smaller
Smaller region,
section. Yeah.
yes.
So it increases, the odds of finding something that's good, even though it can never guarantee that the needle's going to be there. That's one distinction. But computation is really, it's nowadays you need it if you want to be able to rapidly engineer or design proteins.
And I wanted to ask you this question. Have you ever considered applying some machine learning methods into your research through this haystack, or?
Yes, definitely. So machine learning is really powerful when you have enough data to come up with a model that can predict, an outcome to a new set of input, let's say. So in the context of proteins, for example, we have a project right now where we're interested in predicting what would be the best combination of amino acids to catalyze a specific reaction. So if we know thousands and thousands of reactions that are catalyzed by different types of chemicals, can we build a model where we could predict, for a specific reaction, what would be the catalyst that we would need? So that would be an application of machine learning that we're investigating currently.
Oh, that's so cool. Okay. I did a project with machine learning. As I mentioned, it was about deep docking. So it was looking into what types of chemicals could be fit into the protein's
Yes.
pocket. So it's kind of in a similar way, screening through all the database of your chemicals and your reactions to see, well, based on the properties we know and based on their structures, could we predict a model that would provide for a similar protein that we could maybe engineer?
Yes, and much more rapidly.
Yes.
Yes.
It is computationally costly, and it could maybe even lead to some human errors. Do you find yourself bringing some intuition to correct the machine learning's errors, or?
We of course always use our knowledge. Like, for example, it's easy to ask a computer to make a prediction, right? And we're going to get a list of sequences, and if we don't think about it and we just take it as, truth, and then we go and test it, it's probably not going to work that great. So we have to use our own knowledge of what we know of proteins and enzymes to kind of filter these predictions, because there's always going to be errors, right? But the idea is that you just want to enrich. You just want to make sure that within a small enough subset of the possible sequences, some of them may be good. So that's the benefit of computation. We're not at the stage where, we can spit out the one sequence that's going to be perfect, like work exactly as you need it. We're not there yet. But if we can spit out 100 and maybe one of them is good, that's already much better than one out of millions.
Yeah.
Right?
That's true. So now let's get into a little bit more about how the designing of protein works. So could you talk to us a bit about how your lab would design a protein from start to finish? Like, what's the process of finding the idea to your finished molecule?
Right. Okay. So there's, in general, two different types of projects. There's the projects where we start from an existing protein that we try to modify to do something different. And then there are the projects where we start from zero, no protein. So we create a new protein. So, of course, the second option is much more challenging because there's less information to build on. But it can work depending on what we're trying to do. But normally the way it starts is, we have some goal, right? For example, I'll just use enzyme design as an example because that's the most frequent type of project in my lab. So we want an enzyme for a specific reaction. So let's assume that there's no natural enzyme that already catalyzes that reaction, that we would just tweak a little bit to make it better. So we're starting from zero. So normally what we do is we start by generating what we call a thiozyme, which is what would be the theoretical enzyme for this reaction. Like, what are the minimum pieces that I need, which are normally a few amino acid side chains. Now, once I have this hypothesis, I can, using quantum mechanical calculations, I can predict what it would look like in 3 dimensions. Now, from that, I can then see, can I build a protein around this? So I can either take that and try to, stick it on a pre-existing protein. So normally we would have some library of known protein structures, and we would try to kind of docking it, let's say, right? We would dock this thiozyme on each of these scaffolds to see if it can fit. Or nowadays we have new methods where we can build from scratch a whole protein around this this thiozyme. Now, once we have that, we can then optimize the sequences, so predict sequences that would further stabilize this thiozyme. And once we have all those sequences, we filter them. And we filter them based on other types of metrics that we compute that are related to the function we're trying to design. So for example, for an enzyme, we would be interested in what's the interaction energy between the protein and the ligand, let's say the substrate or the transition state we're modeling. Because we want it to be tight. We don't want it to be a bad interaction.
Yeah, you want to make sure that they're like sticking together.
Yes, exactly. Furthermore, we also want to see if they're complementary to each other. Like, if we delete it, does the pocket, is it complementary? How complementary is it? Like, are they binding tightly, or is there a lot of empty space? We can do all sorts of different filtering. And then once we've filtered down the sequences to a number that we can experimentally characterize, we go ahead and we actually make these proteins in the lab, and we evaluate their function. So in the case of an enzyme, we would run an enzyme assay, and we would determine, is it working or not? And the beauty about this is that even if it doesn't work, we know what we've tested. Exactly. So we can learn something. We can say, okay, these ones didn't work. And then we can ask ourselves, why? What did we overlook? But in most cases, we always find something that kind of works, right? It may not be great, but once you have something that kind of works and stuff that doesn't work, then it's easier to start testing hypotheses to make it better.
Right, because you can start building off of it, right? And use it as a base.
Yes. And then at the end, we also do a lot of structural characterization to see whether the structure of what we design matches what we predicted. And if it doesn't, why? And how can we improve it to make it better? So we're always learning something.
When you're trying to modify a protein, what are some of the areas you will mainly target? Because we can't really go and change the backbone, let's say, but how do you decide where should we be making these modifications to make it better?
Right, so it really depends what you're trying to do. Nowadays, there are methods where we can change the backbone.
Oh, really?
Yes.
Okay.
These are newer. Of course, they have their own challenges, but it is possible. But normally, for some function is really easy. Like, well, I shouldn't say really easy, but what I mean is picking the positions you want to optimize is easier. For example, if you're creating a protein that binds some other molecule, you're going to target the binding site. The problem is that we know that for protein function, it's not just the binding pocket that is involved. There's a lot of interaction of effects that can be caused by amino acids far from the binding site, let's say, that could impact the binding. That's a frontier topic or line of research, because it's really hard to predict how an amino acid very far away from, let's say, an enzyme active site is going to make the enzyme better. We don't understand that fully. But that's something that we're very interested in, because we, in my lab, we care about what we call dynamics. So how does a protein move? How does it change its conformation as it's floating around and as it's performing its function? And we know that these amino acids far from the functional site help to modulate these conformational changes. So we're very interested in trying to predict those so that we can make artificial proteins that are as good as natural ones.
Okay. And is that one of the methods you use, multi-state design?
Exactly.
Could
yes.
you maybe just expand a little bit more on that so that our listeners could understand how that works?
Right. Okay. So traditionally, in protein design, specifically in computational protein design, we think of protein structure as a static structure, meaning we know the three-dimensional coordinates of every atom in the protein. And we assume that, unless you're mutating it, they don't change, right? So whatever you're not mutating stays the same. And then let's say you're designing some binding pocket, you're changing those amino acids, so that's changing. But in reality, a protein structure is always fluctuating, kind of like us, right? We're always moving around, our arms, our legs are moving. We're not just like a rigid body.
Mm-hmm.
So in multi-state design, the assumption is there's multiple structures for the same protein, multiple conformations. And different conformations are contributing to different aspects of its function. So what we want is to find sequences that can stabilize different conformations. Let's say the simplest case would be if you think of a protein that would exist in an open and a closed state. So it needs to be open to bind the molecule, but then it has to close so that it doesn't release the molecule right away.
Mm-hmm.
So if I only take the closed structure and I try to predict a sequence that's going to be good for that structure, it may no longer be good for the open structure. So then it's always going to be closed.
I see.
So in multi-state design, we would find a sequence that would be good for both. Open and closed.
So you're taking like both into account when you're designing
exactly.
protein.
So that, of course, adds a layer of complexity because now we're modeling multiple structures. And in our case, sometimes we model hundreds of structures, not just two. So computationally, we need algorithms that can handle all of these different number of structures. But more importantly, we need to know beforehand what are the structures we should be modeling. I mean, open and closed, that's pretty straightforward. But what if there's some other structure, like partially open? Is that important or doesn't it matter? Sometimes it's hard to know.
And how do you decide, what's the thought process when going through, let's say, 100 structures and you're not sure if we should be taking all of them or maybe only 20 of them?
Yes. I guess there's two thought processes. The first one is, I don't know what's going to work, so might as well try them all, right? So let's just see if there's some structure that appears to be favored over the others, right? Or I just want a sequence that can work with all of these. Alternatively, if you have a good hypothesis and you know that it can be open and closed, but you want it to be a bit more closed than open because that's what you think is going to make it bind more tightly, for example. Well, then you can say, okay, so I want the close and the open, but I also want a few more closed conformations that are not the same, but they kind of look closed,
okay.
right? And then you would start designing sequences and testing them. And of course, like I said, we never know beforehand. We only test our hypotheses. And if it works, great. If it doesn't, then we go back and we try a new hypothesis.
Okay. I feel like maybe that's an advantage of starting computationally when being able to design proteins, is that you can test it out by algorithms without having to do a whole experiment.
Yes, although ultimately the ultimate test is the experiment. Yes. But you are correct. I mean, there are ways computationally to eliminate some hypotheses right off the bat. Things that, just don't work. If they don't work computationally, then they're probably not going to work in the lab.
And you've mentioned that you've had multiple of your experiments have partial good results, like working. What's the most shocking result that you've seen in a recent experiment?
Yes, I'll describe this because I didn't believe it at first, like when we started thinking about this project. So our idea was, okay, so there is this particular protein structure that we call a Tim barrel. Okay, so that's, if I was to simplify it really, I would say it looks like a donut. So you have a beta barrel, so it's like a cylinder made of beta strands surrounded by helices. So that fold is the most prevalent in enzymes. So clearly it's a good fold for supporting enzyme catalysis. Other groups have created what we call these de novo Tim barrels, which are very simple compared to natural Tim barrels. So normally enzymes that have this structure, it's not just the donut, it has kind of like a lid on top of the donut so that it can open and something goes inside, let's say, the hole of the donut where the reaction happens, and it closes, right, to hold that molecule in place. But these de novo tin barrels don't have that lid. So
okay, so they're just like the donut bit. They don't have the top part.
The top part, yeah.
Okay.
So it's kind of like saying, I'm going to make a sandwich, but I'm not going to have the top bun. So I'm missing this thing that I need to hold
keep
everything,
inside.
keep everything inside. Okay, but the reason they did it is just to prove that they could create a purely synthetic tin barrel. They didn't care about, making an enzyme out of it. So when I saw this, I asked myself, could I create this lid? Could I add on top of this very simple donut this lid and then create an active site for the reaction that I was interested in? So we developed a computational pipeline to do this, where we basically started by putting in what we needed for catalysis, so the amino acids with a transition state, and then we built a lid that would anchor the catalytic amino acid and close the active site.
Okay.
And then we did a bunch of validation in silico to make sure the protein would fold as predicted. So we used something like AlphaFold. We evaluated whether the interactions with the transition state would be preserved, etc. And of course, at first I'm thinking, I don't even know if the substrate is going to go in. I mean, I have no way of knowing this. I just was interested, could it work? And even if it doesn't work, would the structure exist? Like, would the lid be folded the way we designed it? In any case, so we tested 10 designs, and to our surprise, one of them was active. And then we solved the crystal structure, and it was very, very similar to what we designed computationally.
Oh, that must have been exciting to see.
So that was really exciting. And for me, it was like, okay, I didn't expect it to be that similar to the design model. Because, we had never done something like this before. But that shows you how the field has progressed. There are methods now where you can create backbone elements where you want them. Now, the disadvantage, however, is that there's still inaccuracy, meaning that the enzyme we created, even though it's active and it's the first enzymatically active de novo tin barrel, it's orders of magnitude worse than any natural enzyme.
Yeah.
Right? It's not that efficient. And we think it has to do with the fact that it's hard for the substrate to come in or out of the active site, so that's already slowing down the reaction. And there are inaccuracies in the position of certain active site residues that make it suboptimal for the reaction.
So there's too much resistance for the substrate to go in, like
possibly
as in binding energies too hard to reach.
Yes, so that's a hypothesis.
Yes.
Like, we don't have evidence of this. I mean, we know the structure, but we have the structure in the absence of any ligand.
Oh, okay.
So we don't know how
the
ligand would bind. Yeah,
okay.
Exactly. But that was an interesting result. And now we're expanding upon this. We're using this method. We call this method Canvas. It's a complicated acronym that I'm not going to explain, but we wanted the word canvas because we view this de novo tin barrel as a blank canvas that we can convert to an enzyme for any reaction that we want by creating a custom tailored lid.
Right, because you're like building on it too.
Yes.
So like that's kind of where the canvas part comes in, because it's like you're almost like painting on top of the canvas.
That's right. So instead of creating a totally new protein, we take this minimal protein and we just add pieces to it for whatever need we have.
That's really fascinating to see how science has progressed in that field. And I actually wanted to move along and talk maybe about some applications and future impacts. So if you had to highlight a few of the challenges your work could solve, what would those be? And what areas have you discovered throughout your field where protein engineering could be applied that you did not expect?
Right. So the dream is to be able to make any protein or enzyme on demand for whatever application, and that it works perfectly from that one sequence that the computer predicts. That's the dream. We're not there yet, of course. Normally we have to test, dozens, if not hundreds, to find something that works okay, but probably not good enough for an actual application. And we're still developing proteins for model systems. Okay. Like, I'll give you an example. Nowadays, it's not work from my group, but other groups in the field can relatively rapidly create protein binders. So these artificial proteins created in the lab on the computer that can bind some target protein with, decent affinity, but it's not as good as an antibody.
Oh, okay.
Right?
Yeah.
So, of course, the dream would be to be able to create artificial antibodies.
Yes.
That would bind with that affinity and would have all the other benefits of being an antibody. But we're not there yet. but we're slowly making our way towards those applications. And just to go back to your question about what could be solved, so I'm very interested in enzymes. That's the type of protein that I'm the most interested in. And enzymes could be applied to so many different things. We can use them to make chemicals. So every time you hear about sustainable synthesis or sustainable processes, it means that you're making something without polluting so much, right? And enzymes, they're biodegradable. They work under normal conditions, in aqueous solutions, normal pressure, room temperature, etc. So they're synthetic processes that use enzymes as catalysts tend to be much more sustainable than other types of processes that use different catalysts that are made by humans, basically. And then, there's also medical applications. You may have heard of CRISPR-Cas9.
Yes.
So, that's an enzyme, the Cas9. So people are engineering these enzymes to go and cut more efficiently a specific DNA sequences so that they can hopefully cure some, genetic diseases. And so there's been a lot of work in that area as well.
I was actually going to transition into sustainability because we know there's a lot of toxic catalysts. We're always talking about how can we improve make the earth greener. So you talking about developing sustainable biocatalysts to make greener applications of things are interesting. Have you had a project towards that?
Yes. So several years ago, we engineered an enzyme. So it's a natural enzyme that normally makes D-alanine. So it's an amino acid, but the D version is the mirror image version. Mm-hmm. So it's the enantiomer. And there's a bunch of applications for these types of, D amino acids in natural products, pharmaceuticals, peptides, etc. But they're hard to make chemically because normally, chemists would make a racemic mixture, so a mixture of the D and L, the two enantiomers, and then they would have to separate them, which is not that easy. But enzymes, they'll make one or the other. They're super specific. So our idea was, well, can we engineer this enzyme that makes D-alanine, but make it produce a different D amino acid that it normally doesn't make? And the one we targeted was D-phenylalanine, which is a much bigger amino acid. So the idea was we go from a very small one, D-alanine, to a much bigger one, D-phenylalanine. Mm-hmm. So our hypothesis was, let's just make room in the active site. Let's just, mutate amino acids in the active site to smaller ones so that we can make more space. And then we'll test those mutant enzymes with the molecule we want to see if it works. So it ended up working. And then in collaboration with a group in the UK, we developed a biocatalytic cascade where this enzyme was mixed with other enzymes so that we could produce D-phenylalanine starting from a much simpler small molecule, fully in a biocatalytic process, meaning that catalysis was exclusively carried out by enzymes. There was no small molecule catalyst. And that worked well. And eventually I got an email from Merck, who's one of the
big
very big pharmaceutical company who was inquiring about the possibility of using these enzymes in the synthesis of some compound they were interested in. Of course, it didn't mention what that was. But then we licensed those enzymes to a company that would make them and sell them to Merck.
Wow. Yeah. So there are immediately direct applications, and that's very interesting to see.
Yes, it's just that it's not, at least at this point, it hasn't been applications that, everybody would have heard of. Mm-hmm. They're still specialized. Yeah. But, little by little, these things, get used.
I wanted to ask you, when you first got into protein engineering, what are some breakthroughs you wanted to see as someone who was starting into the field? And looking now, after years into the field, what have you seen accomplished?
Oh, yeah. Many things. Yes. What I can mention, so already you guys as undergrads are learning things that did not exist when I was an undergrad.
Could you give us an example
of
one?
Yes, I'll give you the example. It's not directly protein engineering or design, but it totally has revolutionized protein design. And that's the development of AlphaFold. So when I was a student, of course, people would always say, we know that an amino acid sequence will give you this one protein structure, right? It'll fold. But we don't know how to predict that. Mm-hmm. Like, I cannot, from the sequence, predict the structure. That was a grand challenge that had been around for decades. turns out that that was solved, for the most part, in 2020, where this company, DeepMind, developed a AI method to basically predict the structure of a protein from its sequence. And when I heard about this the first time, I'm like, okay, that can't be true. I mean, it's not going to work all the time, right? And it's not perfect. I mean, some sequences it still struggles with. There are errors in let's say, the details, but for the most part, you can, very accurately predict the structure of a protein from its sequence using this tool. And now this is a tool that we're using in protein design because when we design a new protein, we evaluate its predicted structure using AlphaFold. So if AlphaFold tells us, okay, the structure I predict matches what you designed, then it increases the likelihood that this could work. It doesn't mean that it will, but it increases the odds. And in terms of the new developments in protein design, this is not the work from my group, but from others. They understood that if AlphaFold can take a sequence and predict the structure, we should be able to do the opposite. Take a structure and predict sequences, because that's what protein design is.
Yeah.
Right. So that also, those developments led to the Nobel Prize last year in chemistry.
I've heard about that.
Yeah.
Actually, in my protein class, I don't know about you as well, but
I think, briefly, Dr. DaCosta might have mentioned it, yeah. So do you think as AI progresses then that we're going to see like a more of like a bigger increase in like the computer's ability to like estimate the function and like structure of protein?
Oh, definitely. Yeah. So it's just a question of time because the more data there is and the more the algorithms become powerful in terms of using this data to build really good predictive models, it's just going to get better and better. The problem that we have to keep in mind, though, is that there are still many things we don't understand. So even though AI could possibly predict something that's going to work, it doesn't mean that we'll understand why. So there's still going to be work for scientists to try to understand things. That's never going to go away. There's always going to be cases where the AI cannot do it. I mean, you probably have used, ChatGPT or these other types. You know that there's hallucinations, that there's biases. So same thing in protein design. The AI will overemphasize the most common things. Like, for example, when we use these AI-based methods to design new proteins, it's really good for alpha helical proteins, but it's not so great for loops or beta strands, just because not only are those harder to predict, but there is fewer examples because these structures are less, let's say, repetitive. Whereas a helix is always a helix, and there's so many helices in the PDB that it's really good at predicting this. But loops, that's still very hard. And a lot of protein functions depend on flexible loops. For example, antibodies. That's one of the reasons why it's so hard to predict, in silico an antibody for specific antigens, because the binding interaction is caused by these flexible loops, which, are very hard to predict.
That's true.
All right, so in terms of advice for undergraduate students, you've already done your undergraduate degree, so you've got a lot of experience to share. Do you have any key pieces of advice that you can share with future students?
Yes, so I definitely have advice, and I'll divide it in two categories. There's advice from a scientific perspective or learning perspective, and then advice from more of a personal or attitude perspective, if you want. So from a scientific perspective, if you want to do research, what's very important is that you build your base of knowledge, so the fundamentals. So if you're in my field, let's say protein design, that involves biology, chemistry, and physics, right? Because biology is understanding protein function. Then chemistry is understanding protein structure and how that relates to function. And in physics, that's the basis of everything, but basically understanding thermodynamics and kinetics, that impacts how the protein behaves. So if you have a strong foundation, it's easy then to expand into new areas, right? Furthermore, the other thing that's very important that I think often is overlooked is learning to code or program, or at least be able to read a script and understand, computer languages. Just, for example, using Linux. understanding the logic of typing commands versus, clicking on things. It just helps understand how a computer works, and then you can more easily use it for what you're trying to do. So that's what I would say in terms of the learning or science part. And then from the personal aspect, I would say, often as an undergrad, it's hard to connect with research, right? It's like, oh, the professors, they're so distant, or I don't know enough about this, so how do I go about it? What I will tell you is that if you're really motivated and enthusiastic, people will want to work with you, right? We professors used to be students. We work with students. We like to work with people who are as enthusiastic as we are about certain topics. So if you are super enthusiastic about something, you just need to find a person at the university that studies that. And if you're really interested and you reach out to the person and you show them that you're really interested, I'd be surprised if they wouldn't be interested in talking to you. Because, we're working together as a team. Like, for example, in my lab, the students do the experiments. I'm not the one working in the lab anymore. But I'm super enthusiastic about the research, and I love it when they do an experiment and they show me, because I'm not the one doing it, but I want to know the outcome. So then I share that passion with them, and all professors are like that, right? So if you're really interested and you contact the person, you demonstrate that you're really interested, and kind of like what I told you earlier, when you reached out to me, you had prepared, your explanation of what it is that you're trying to do, and I could tell that you had really thought about this. That made me enthusiastic to come to talk to you, because I could tell that you were going to be interested. So the advice for the undergrads is don't assume that there's no opportunity, right? You can make it yourself, or you can join, let's say, an iGEM team, or you can try to go for one of these scholarships where you can work in a lab for the summer. And even without scholarships, you can reach out to professors, and if you're interested and they can tell, they're going to be interested to talk to you.
That's true. I agree that most professors, we're in a world of learning and knowledge. We all want to share that knowledge.
Yes.
So most people would be open to talk. And I really appreciate hearing that from a future professor I might take as well. So
Yeah.
Thank you so much, Professor, for coming in and talking with us.
Yeah, we really appreciate it. And it was really interesting to hear about what you're working on.
Thank you.
Thank you
And
so
remember, many years ago, I was one of you.
Thanks so much for joining us, Dr. Chica. We really appreciate discussing with you how building new enzymes can bring science and creativity together.
And thanks to our listeners for tuning in. I'm Lilia.
And I'm Amelia.
And we'll be back next time with more stories, insights, and a fresh cup of tea to spill.
Remember, the journey doesn't stop here. Keep thinking, keep questioning, and keep steeping in inspiration. See you next time.